University, where I get to work with his wonderful team as well. Now, so what excites me? I care very much about cardiac lesions. So the most detrimental lesion is following ischemia. It was mentioned earlier that my main area of research looks at cardiac reperfusion arrhythmia mechanisms. This is a huge story that I won't go into today. Instead, I'm going to talk to you about another one of my projects. The focus is on the most prevalent lesion, so atrial fibrillation. This is the most common type of arrhythmia. It affects millions of people. We intervene with ablations, and this presents a very high economic burden. So we're going to focus here today, and I also want to quickly mention that the work that I will show is actually a sub-project within a massive collaborative research center in Germany, the CRC 1425, where the speaker is my mentor, Professor Peter Kohl. And if you have questions about my project and related projects, please don't hesitate to contact us here. Okay, so what should the heart normally look like? So during sinus rhythm, under normal conditions, this is what you would expect from the heart. So you would expect from this cartoon that you have normal electrical activation driven by these pacemaker cells. When looking at mechanics, this here is an MRI showing normal mechanical deformation of this four-chambered organ. And then also when looking at the open chest of a pig, you see the atria is at the top, the ventricles, and how it should normally beat. This is wonderful and great, but what happens with patients with atrial fibrillation? So here you get aberrant electrical conduction in the atria that so abnormal electrical impulses. I mentioned it's the most common type of arrhythmia. These faulty electrical signals are very fast. You can see it in the video here. They're irregular, they're disorganized, and often the atria will get out of sync with the ventricles, which can lead to the heart not pumping as efficiently. Now, sadly, with these patients with atrial fibrillation, the saying is AF begets AF. So it's a pretty bad situation, and these patients, of course, need help. What can we do? So with these patients, the second line of therapy, or actually more recently, the first line of therapy that's being recommended is some form of catheter ablation. So here, a cardiologist, let me put my pointer. A cardiologist will get venous access to enter the atria. They introduce a mapping catheter to get electrical and anatomical information from the subendocardium. The cardiologist will then introduce an ablation catheter where with radio frequency, for example, they can apply these heat lesions in the atria that aims to eliminate either the triggers initiating atrial fibrillation or the underlying arrhythmogenic electrical or mechanical substrates. So in the end, these patients with atrial fibrillation have these scars that are meant to deal with these aberrant electrical conductions that are not desirable in the heart. I also want to have you think about the cells that are present in the heart. So when our colleagues do single nuclei RNA sequencing, they show very clearly that we should shift away from this cardiomyocyte-centric view of the heart. This is not the main cell type in the human atrium. There are many more fibroblasts, there's tons of macrophages, there's neurons, endothelial cells, tons and tons of other non-myocytes that are at play. These non-myocytes are very important because firstly, I mean, after an ablation or a lesion, I mean, where we reduce or eliminate cardiomyocytes, these are the main players that remain. They are interesting because, I mean, in part, Alex Quinn has previously shown that when non-myocytes are labeled with a membrane potential reporter, shown in green, he has and others have shown that there's cardiomyocyte, non-myocyte electrical conduction in the scar border zone. So this is wild and wonderful because firstly, I mean, these non-myocytes that remain in the scar are not just inert cells. I mean, they're very active. And also I'd like to have you think of the scar, the lesion, as being very much alive. So this is not just dead tissue. Now, so what? What's the problem? So you have these patients with atrial fibrillation, they undergo ablation therapy. What is the problem? So the problem is that these atrial ablation lines that are put in place become electrically transparent over time. And so the first time ablation success rate is anywhere from 20 to 50% and can rise to, but rarely exceeds, an 80% ablation success rate upon repeat ablations. We hope to be able to do better. Why do these lesions become transparent? Why aren't they durable? One mechanism that we think is that there might be an involvement of cardiomyocyte, non-myocyte interactions at play that need to be investigated. And so the overall goal of this study, this project, is to try to increase first ablation success rate to get a better scar. To get this better scar, our idea of a nice lesion is one that's narrow, that's transmural, continuous, permanent, and ideally cardiomyocyte specific. But to make this wonderful ideal lesion, I mean, we need to understand the scar, which I hope I will try to show you what we've been doing here. Now, to try to understand the scar or to steer cardiac electrophysiology, I mean, I don't have human volunteers, so we need a model to study this. And I would argue that for this study, the best model is the pig. Not only does it close the translational gap between the clinics to basic science, it could also be used to reverse translate, so to find problems or observations in the clinic and to bring this back to the bench side to understand mechanisms and to find solutions. Of course, with any model, there's advantages and disadvantages. A key advantage for us would be instrumentation. So we're using standard clinical approaches and equipment that we use in humans in the pigs. So by using a smaller mammal, I mean, our tools are just simply too big. Another thing about the pig is that it closely resembles human cardiovascular anatomy and hemodynamics and electrophysiology, which is very important. There's a growing number of transgenic pig models that are available, but with advantages of a model, there are disadvantages. The big ones being that these are very expensive experiments, unfortunately, and they're very time and labor intensive. To show you just how crazy these days look like, so when I'm coordinating a pig day, I mean, it's very collaborative. So it involves an army of people from anesthesiologists, veterinarians, cardiologists, cardiac surgeons, radiologists, scientists, so many people that come together to make this day possible. So you see this quick time-lapse video of us in the operating theater, where we're doing all our in vivo experiments. Beyond just the operating theater, after we've inserted our lesion, we keep our pigs alive for 8 weeks, we remap, and then we post-process in the lab. So again, you see us busy bees in the lab. Tons and tons of scientists come together to do a battery of structural and functional analyses. So this here is a quick overview. So we're working with German landrace pigs or Munich hybrid mini pigs. Day 0, we are doing our in vivo mapping before and after ablation. And then 8 weeks later, we again remap, sacrifice the animals, and then we see what happens with much more resolution in the lab. Okay, and now to organize my talk, I mean, there are many things that we've done, but to kind of guide you through, I basically will present what we've done according to scale. So I'll start at the organism level, then move down to the organ level, tissue, cell, and so on. When trying to understand these lesions, we try to understand them in vivo first and then ex vivo. And we're also trying to characterize the changes in electrics, mechanics, and structure. And we have various tools to help us understand this. Now, all of this work then goes back feed into our computational modeling for further hypothesis generation. Okay, so let's dive right in. So to understand what's happening in vivo, the changes in electrics, here we perform electroanatomical mapping. So this is a routine technique used in the clinics. Here is the right atrium. You see the superior vena cava up here, inferior vena cava, 2 views of it. And you see on your baseline, before we ablate, you have normal sinus rhythm. So you have normal voltage. Here's a profile of high voltage areas, low voltage areas, and very low voltage areas that we characterize as control. After we get this profile, we then ablate, so we introduce our scar. So we're targeting the posterior wall of the right atrium from the superior vena cava to the inferior vena cava. And you see then we get a reduction in voltage, as you would expect, and then more low voltage areas. So reduction of high voltage areas and an increase of low voltage areas, and slowing of conduction around the scar. This is great. And then after 8 weeks, we let this lesion develop, we remap and assess it, and we see that the lesion is comparable to immediately after ablation. So this is great. Not only do we aim to understand in vivo electrics, but we also want to understand in vivo mechanics and structure. So then another routine approach that we use is magnetic resonance imaging in collaboration with our radiology colleagues. This is our pig heart. You see that it beats normally. That's not an ideal view to look at the area that we target. So here's a transverse view of sections going from the cranial to the caudal end of the heart. So you see the beginnings of the superior vena cava and then the atrium. And this is the area that we ablate after measuring what it normally looks like. 8 weeks later, we assess this lesion in vivo as well as ex vivo. Here's an example from sagittal sections where you see this white area of where the lesion is located. And we can also see this visually when the organ is extracted. Speaking of the organ, as we move down in scale, so here at the organ level, trying to understand electrics, we can perform optical mapping experiments to get information about whole heart electrophysiology. Here we Langendorf perfuse the pig hearts. This is what that looks like. We perfuse with a physiological solution. We load these hearts with a voltage sensitive dye. And then we can measure optical action potentials across the entire epicardial surface and look at changes in electric dynamics. So in much higher spatiotemporal resolution. So now moving further, getting down to the tissue level, we can now dissect the heart. So here you see us separating the atria from the ventricles, and then we divide the right atrium from the left atrium. Here's the right atrium, where we then also open up the superior and inferior vena cava. In pig, this is actually called cranial and caudal vena cava, to flatten out the posterior wall, which is where we've targeted and ablated. You see the location of the scar. We can characterize the scar. So this is the native tissue, and from this strip, you can see the healthy pink area here compared to the scar tissue, which it has this nice transmural scar. We can characterize fibrosis with histology in the scar area compared to healthy area. We can do immunofluorescence labeling. So here again is the strip where you see the healthy area here versus the scar area and then the border zone. If we zoom in at the border zone and I label cardiomyocytes in red, non-myocytes in green, and connexin 43 in purple, what you'll see that's very interesting for us at least is the presence of this connexin 43, this gap junction protein between a non-myocyte and the cardiomyocyte, suggesting that these cells may be interacting. Another thing that we can do in collaboration with Joseph Madel is this label-free second harmonics generation imaging. So here we look at collagen in the healthy area compared to scar area, and you see that this collagen is very curly and disorganized compared to control. So tons of structural analysis that we can do. An important note is that firstly, I mean, it's a pretty massive scar. So here you see its location in one pig, but also, I mean, while we always target the same region, so we're trying to get that posterior wall from the SVC to the IVC, depending on the day, the pig and the cardiologist that's available, the scar ultimately ends up looking very different. So repeatability is an issue. Now, still at the tissue level, when trying to understand mechanics, I can pass off some of these samples to our biomechanics team at our institute, and they have this wonderful nano indentation technique that allows, so there's this optical fiber that can measure deformations of this cantilever as it approaches a sample. This is a very sensitive technique to give us information about tissue stiffness. And so here, when you look at my sample of the pig scar healthy area, when they perform these line scans in the scar area and a remote region, they certainly can measure that the scar is stiffer. My colleague Teresa can do more work with these biopsies. So she has a lot of experience, but also rabbits and humans. So she gets these tissue samples, she can agarose embed them, she can slice them further into 300 micrometer sections, and then she can put them in these wonderful biomimetic chambers, applying a load to keep these slices in culture for several days. So she shows that these cells are contractile over several days, they maintain key functionality for up to a week. Beyond the tissue level here, we can go down to the cell level. So here, my colleague Joe has tons of experience with enzymatically digesting these cells in slices to do single cell experiments. He shows that these are pig cardiomyocytes are calcium tolerant. They have these beautiful striations. They are paceable, and also interestingly, with the pig cardiomyocyte, you'll note here that it's multinucleated. And then my colleague Anna can do more characterization, where she can patch clamp atrial and ventricular cardiomyocytes to try to characterize healthy and scar tissue or cells. Now, we do a lot more work, but ultimately all of these experiments feed into our computational modeling. And here, what I show you is an example of a 1D strand of 15 cardiomyocytes in red, electrotonically coupled to fibroblasts. And so in the mouse, what you can see is that we get this passive conduction of atrial signals through non-myocytes of up to 4 fibroblasts. When using another species like the rabbit with an action potential that's actually more similar to human, you can, especially with its plateau phase, you can see that we can get, we can extend this bridging of passive conduction of up to 13 non-myocytes compared to 4. This here suggests that actually all of our work with small animal models will underestimate the significance of this passive conduction in a more human-like situation. So that's why the pig is very important. And when thinking about ways we might be able to modify or steer the lesion, some ideas we have would be to, for example, reduce connexin. So here, if we reduce connexin by 30%, you see that we can limit this bridging, or if we mildly or significantly overexpress potassium channels to hyperpolarize these fibroblasts, we can also make that scar more durable and prevent these reconnections that we have to test experimentally. Some additional surprises that we've been finding along the way that are totally unexpected is just the high incidence of pericarditis. So this is a Langendorf fused pig heart, normal, and then in this case with severe pericarditis. So this is a form of sterile inflammation in the clinic. It's reported at about 10%, but with our animals, we actually see this in more than half of our pigs. We think this is underreported because it's actually very hard to see pericarditis. So when we look at our MRI data, electroanatomical data, echo, whatever, I mean, it's not obvious to us either. So it's likely underreported. And then another interesting preliminary thing that we've been seeing is just the presence of melanocytes in the scar, or melanocyte-like cells in the scar. So why should there be melanocytes in the heart where the sun does not shine? I mean, I'm not really sure, but when we characterize this very preliminarily further, looking at the healthy area, we see that there isn't the presence of TRP1, which is a marker of melanin synthesizing cells, but then when looking at the scar area, we do see this. What role might it play? I mean, melanocytes are thought to be stiffer, so maybe this is a mechanical substrate for arrhythmias, but this needs a lot more work. But in summary, I hope that you were able to get a little flavor of all of the exciting and wonderful things that we do in Freiburg using this large translational pig model. A few take-home messages, I mean, are first, I mean, it's hard to interrelate the lesion. So when looking at the MRI data, electroanatomical mapping data, echo, or all of the structural things that we do ex vivo in the lab, I mean, with all of these different imaging modalities, we get different information about the lesion. Secondly, I mean, it's important to note that there's a problem with lesion reproducibility. So while we are always aiming to ablate the exact same way, no two scars have been the same. And that scar by radiofrequency is still actually quite large. So we would like to be able to still further limit how much we burn and without the risk of getting reconnections. In terms of non-myocytes, I mean, there's tons of them in the lesion. I think they're highly involved in atrial lesion repair. And if you take away nothing from this talk, I mean, especially to my clinical colleagues, I hope that when ablating, you can think more, that you can appreciate that it's not just about going in with your catheter to ablate a single straight line. There's tons of non-myocytes at play, and the lesion itself is a living thing. In the future, I mean, we're still actively trying to steer post-ablation lesion properties in the pig to make better scars, but to change the scar, I mean, we need to understand the scar. I told you a lot about our work with radiofrequency ablations and how we're trying to understand this type of lesion. We're also trying to do this in after cryo ablation, which is a common alternative to RF. But actually, in the end, to truly reach this ideal lesion, we think the best way would probably be a surgical approach. So here, we think that we would get a much narrower lesion that's transmural, continuous, and permanent. So we'd like to revisit the classic cut and sew maze surgical approach. We're doing this in mouse. So here, the right atrium is clamped. We cut the atrium, and then my colleague Lewis has these nice micro suturing techniques where you can close up this lesion, and we can try to understand this type of scar. Another type of scar that we're trying to explore is one that there's a lot of buzz in the field, so pulse field ablations. So here there's high intensity electrical pulses that are applied. This creates permanent pores in the membrane, and as cardiomyocytes are more sensitive to these high intensity electrical pulses, it's thought to be a cardiomyocyte specific approach. It's also, I mean, there should be less undesirable damage to like nerves or the esophagus compared to the classic radiofrequency cryoballoon ablation approach. And very early reports show that with this pulse field ablation, there has been a higher first ablation success rate. But by going to some meetings, I mean, while this is very exciting and we're actively exploring this and going forward, we don't understand the mechanism by which this works, which we want to further explore. And also these lesions at the moment are quite massive. So this brings me to the end of my talk. I, of course, have tons of people that I need to thank. So the entire Institute for Experimental Cardiovascular Medicine, tons of scientists and a wonderful team that have worked tirelessly on this project. I also have to thank our animal facility, our cardiologists, our industry partners, surgeons, and many, many others that I can't even list on a single slide. I'd like to thank our funding, and at this point, I'm happy to take your questions.
Okay, thank you so much, Dr. Chleilat, for such a great talk. So we will move into our Q&A session now, at which time I will be ending our recording.